Now I invite all of the speakers, Professor Amin Subandrio and Dr. Smith and Dr. Neny, to be seated in the available seats here. And we will have 15 minutes discussion until 11 o'clock. Because today is Friday, we have a Friday praying time. So I open 3 questions first for this opportunity for this discussion. Okay, then I will ask first. Huh? Okay, so you, one, two, one more. Okay, please, you may start. To whom, and please indicate your name and university from.
Thank you for opportunity. I'd like to address my question to Dr. Smith about EPV vaccine. So in your presentation, you mentioned that the main characteristic of EPV infection is that they persistently infect B cells in a latent state, meaning that they have many strategies to avoid immune systems, such as very low antigen presentation. Since your vaccine is targeting LMP1 and EBNA, do you have any information of data about how high the level of expression of LMP1 and 2 or an EBNA in a EPV infected cell? Since that if the expression is very low, then your vaccine is very low in their efficiency. If the expression is very low by EPV infected B cells, do you have any system to increase the expressions of LMP1 and EBNA in vivo, so then it is more likely that it will be targeted by the polyfunctional T cells? And then the second question is about the factors. Since you choose adenoviral factors for your vector delivery system, I have one question about why do you choose the adenoviral factor in your system? Because in my knowledge that the adenoviral infection is very prevalent in community, meaning that we have pre-existing immunity against adenovirus that can decrease the efficiency of the vaccine. So maybe you have any comment on that. Thank you very much.
Okay, I'll start with the second. That's true that pre-existing immunity can be a problem for adenoviral vectors, but I think there was some studies with you looking at HIV vaccines with these strains, and you could induce strong CD8 T cell immunity with human adenoviral vectors, even despite having pre-existing immunity. But there's also groups developing actually adenoviral vectors from chimpanzees, for example, as a means of delivering, using a chimpanzee adenoviral, putting in antigens. There's been, I think, even hepatitis C type vaccines. So adenoviral vectors do still have potential use, but there are some complications with pre-existing immunity. So yeah, the other question, that is the ultimate difficulty in determining the efficacy of this type of approach, where if we can prime, despite the fact that we can generate LMP1 and EBNA1 specific T cells, can they actually recognise the virus-infected tumour cell? So we know for EBNA1 that it's work that a lot of work that's actually come out of our group from other senior researchers within our group have shown that these proteins are very good at regulating their own expression, and they are expressed at a very low level. So this will make it hard for the immune system to see them. But actually what we've also seen for LMP1, this is not actually the case for LMP1, but it is still very poorly immunogenic. Even at high expression levels, we have difficulty seeing it. But that's probably not the case for LMP2. So we think we can get reasonable targeting of LMP2. Now there are mechanisms which we've looked at to enhancing EBNA1 expression. Now this is very specifically to do with the way that the protein works. It's quite complicated to explain, but I don't see whether that they'll actually be applicable in this setting. Really, it's going to be one of the difficulties, and it's why the virus is obviously so successful, is that it can infect B cells and it can turn itself off and turn itself on, express a very few numbers of proteins. But we hope at least by trying to improve the quality of the cells, maybe they'll be able to recognise that antigen which is at low density. But that's something we need to see, yeah.
Okay, thank you. If you haven't satisfied yet with your questions, you may continue the discussion later on during the break. And now I invite the second Yeah, please.
Okay, my name is Wahyu Hidayati. I'm from University of Muhammadiyah, Prof. Dr. Hamka, Jakarta. I would like to ask question and just commentary for from the first until the third narasumber ya, speakers. Sorry, for Professor Amin Subandrio, I interested in dengue research, and you say that in the past, dengue research already promised it will have vaccine dengue, and it is failed. And now we have consortium. The question is, what is the obstacle or difficulties in the last project for dengue vaccine? And then that's for Prof. Amin Subandrio, and then for Mr. Smith, I learned about immunology. And I'm curious about immunotherapy. Fortunately, you give more knowledge for me about immunotherapy, especially in cancer. I just want to ask you about what kind of immunotherapy it is. Can it be used immunotherapy for genetic cancer that caused by genetic disorder, and then what kind of antigen that efficient for cancer viral therapy? It is for T cell or B cell more efficient. That's for Mr. Smith, and for Miss Neny. It is a good way for Indonesia to make our own factor for Hansenula, because we know the Pichia pastoris, this factor and bacterial factors we are imported, and perhaps your team work now, it will make us as biotechnology scientists more easier to make another protein recombinant, and it make us more not easier. We use less of money because we don't have to import again from outside the countries. Okay, thank you very much.
Okay, time for you, Prof. Amin.
Thank you very much for your question. So if we are talking about dengue vaccine, it's the approach is a little bit different with other vaccine, because dengue infection we have to look at carefully to the pathogenesis of the disease. Dengue infection not only cause fever, but in severe cases they could develop to more severe disease due to immune response of the host. So as we understand that dengue virus has 4 types, and we have to be successfully stimulate the development of antibody to those 4 types of dengue virus, because if only 3 types, serotypes is protected and not the other one, then there will be, of course, there will be a cross protection, cross reactivity. The dangerous is if we have antibody with a level of sub-neutralizing, then there will be a danger in establishing the severe immune response, which could cause hemorrhagic, dengue hemorrhagic, and then also dengue shock syndrome. That's why those who are interested in developing dengue vaccine should consider those principles. So we have to establish protection to those 4 serotypes at the same level. So there should be enough protection to those 4 types. That's the main constraint of establishing dengue vaccine. That's why we are already in 30 or 40 years in dengue vaccine research, but still not a single candidate is satisfactory. up to the moment. Thank you.
We need to go?
Yeah.
Can you hear me? Okay, so I think the second question was about the type of immune response we're targeting with immunotherapy, and it's primarily looking at generating T cells. It's really cytotoxic T cells that we primarily aim to, and most of the immunotherapies are targeting. I could actually probably talk about immunotherapy for an hour, and the different mechanisms that can be used.
No, the question is about the immunotherapy for non-viral.
Yeah, that's where I'm getting to. That's where I'm getting to. Okay, so that's what I was about to say. There's actually lots of avenues that people are using to develop immunotherapeutic approaches for non-viral associated cancers that might be genetically associated or associated with changes. So the type of approach we use is being applied in those sort of strategies where people will take cells out of the tumour, and then they think they're actually specific for mutated epitopes or mutated antigens in those tumours, and then they'll reinfuse them. The real breakthroughs in the field of cancer immunotherapy in the last 3-4 years, and it was actually breakthrough of the year in science last year, these antibodies that essentially work by activating a T cell response. But what they block is the regulation of the immune system. They block these molecules that regulate your immune system. And these are antibodies that have been developed and have been shown to be very successful in diseases like melanoma. And I think the way they work is that they release the brakes on your T cell responses that would recognise what are normally self-antigens. But they can have, obviously, some actually fairly significant side effects, which are associated primarily with autoimmunity. Because if you're turning off all these mechanisms that control your autoimmune response, you develop autoimmunity. So there are a number of different ways that people are developing immune therapies to treat non-virally associated malignancies. Yeah.
Thank you. Dr. Neny?
Thank you. Thank you for the hope for the factors and expression system. For the factors, actually, it's not so challenging that if we compare to the host strains, because we can design the factors based on the information of the genes, and then we also send to the vendors that will make the synthetic gene of these factors. But for the making the strain of the Hansen polymorpha is quite difficult also, because it's very big organisms, and we already have the strain that have the marker negative for the marker of the ura or ura gene negative, but we have to test the stability, because in several clones we know that we observe that this ura genes is the yeast is not stable, so we have to do several clone selections. And the other things that we don't explore in this consortium is the potential of yeast expression system from the yeast, Indonesian yeast, because a lot of Indonesian yeast in Indonesia right now, the isolate is in LIPI, but no one of the Indonesian biotech use for the this system for expressed or making the recombinant protein based on our Indonesian isolates. Thank you.
Thank you very much. I think we still have, I'm sure there are still many questions will be arisen from you, but because time is limited, so we have to stop in this time, and we will continue the discussion during the break, if you may still have, or if you need more information, we would like to ask you your email address, so you can email directly to these three experts. So please give big applause for these three speakers. We have a very excellent presentation, very excellent information, and also and discussion. Yes, thank you. Now I'll give it back to the MC for the next agenda, and I think, yeah, okay, please.
Okay, thank you for all of the speakers and also moderator. For all of the speaker and moderator, please stay on the stage. And we kindly ask Professor Sofia Mubarika to give souvenirs to all of the speakers.
Okay, thank you once again, Professor Dr. Sophia, Dr. Amin, Dr. Corey Smith, and Dr. Neny. Okay, ladies and gentlemen, so we have now arrived to the end of this first session of the presentation. Before we proceed to our lunch session, we will have the surprise for you, because I can feel that many of the participants are feeling sleepy. So we have the surprise gifts for you. Okay, so we will be having the 3 questions from the 3 presentations that already took place on the stage. So the participants who will be answering the question, they will be getting the surprise gifts from us. So it is to check your attention, how attentive you were in this presentation. Okay, so are you ready? Please scream yes. Okay, that's good. Okay, the first question is from Professor Amin, and the participant who will be answering this question has to scream yes and raise the hand. So who will raise the hand and say yes, he will be given the opportunity to answer the question. Okay, so here I go, okay. We start this questioning session now. From Professor Amin. The question is, which one is not an infectious disease? I repeat, which one of the following is not an infectious disease? And the options are A, dengue hemorrhagic fever, B, diabetes mellitus, and C, diphtheria. Okay, I will count 1, 2, and 3, and one who will say the yes, he will be given the opportunity. Okay, here I go. 1, 2, and 3. I think the lady with the blue scarf was the first to scream. This one, yes. Can you please come in the center and say the answer?
The answer is option B.
What is option B?
Diabetes mellitus.
Okay, yes. Is that correct? Yeah. Okay, and now the time for the second question from Dr. Corey. What are the EBV proteins expressed in NPC, nasopharyngeal carcinoma? I repeat the question. What are the EBV proteins, Epstein-Barr virus proteins, expressed in nasopharyngeal carcinoma? So the level is getting higher. Okay, once again I repeat the question. Oh, one protein. Okay, just one protein, okay. So anybody? Okay, please ma'am, please come in the center.
LMP1.
Is that correct?
And
okay,
no.
Please give a round of applause for her. Very good. Okay, and now the time for the third question. Okay, get ready, please. Okay, the question from Dr. Neny. What are the 3 key elements for the production of recombinant vaccines? Okay, so there is no need to repeat the question again.
The first one, we have to find the antigen, and then we sequence it, and then we put it into the vectors. After that, we clone it into the bacterial or the cells. Yes, right, the key?
Is that correct, Dr. Neny? Yes, very good.
Thank you.
Thank you. For the prizes, I would like to request Professor. What is his name? Professor, please come front and please distribute the prize. I don't know. Okay, can we have the participants who answered please come forward? Okay.